So let's dive into amino acids. So first we tell the classes of amino acid. So first of all, the gluconeogenic amino acid. So the mnemonic for gluconeogenic, I met his Valentine and she is so sweet. So met or methionine, his histidine, Valentine or valine. Then we have the ketogenic. We have two ketogenic, the two L's, the leucine and the lysine. Then we have the acidic component. So we have two acidic, glutamic acid and aspartic acid. Regarding the basic, we have histidine, then lysine and arginine. Then we have branch chain amino acids, most common in bodybuilding and profession. So branch chain amino acid, we have the valine, the leucine and isoleucine. The next we have the essential amino acids. Then for the essential amino acid which are very much required for our body are the P, V, T, Tim, Hall. So for P we have the phenylalanine, and this phenylalanine we will discuss it in the catecholamine synthesis. So this phenylalanine, it give tyrosine give dopa, which gives dopamine, and which gives epinephrine and norepinephrine. Then we have the valine, the threonine. Then we have tryptophan. So tryptophan, if it uses B6 and B4, it gives serotonin and melatonin. And if it uses B6 and B2, it gives niacin, giving the NAD. So this tryptophan is very much important in albinism, in serotonin syndrome, in alcoholic NADH problem, in pellagra. So this tryptophan amino acid is very much important in these four type of scenarios. Then we have the isoleucine, the methionine, and then histidine, which uses B6 to make histamine, and this histamine is used in hypersensitivity reactions. Then we have the leucine and lysine. Another derivatives of amino acids are the glycine. Glycine gives using B6 in the porphyrin heme synthesis. Then we have arginine. Arginine using B4 give nitric oxide, used in pulmonary hypertension with dilation. It gives urea and creatine and urea, which we will discuss later on in urea cycle. Then we have the glutamine, and this glutamine gives GABA and glutathione. So this is all about the amino acid derivatives. The next one is the urea cycle. So in the body uses the amino acids and produces ammonia. And this ammonia, how this ammonia is converted into urea and excreted from the body, we will discuss here. In the urea cycle, in the mitochondria, we have ammonia plus carbon dioxide in the mitochondria. It uses N-acetyl glutamate, which is the activator of the synthase enzyme. What synthase? Carbamyl phosphate synthase. which is a rate limiting enzyme. So it gives the synthase give carbamyl phosphate. This carbamyl phosphate is ready to transport out of the mitochondria by reacting with ornithine. So ornithine and carbamyl phosphate, they make up and they use an enzyme known as ornithine transcarbamyl enzyme ACE and get out of the mitochondria to give citrulline. This citrulline, using the ATP molecule and enzyme synthase to make arginosuccinate enzyme, arginosuccinate, and using aspartate as a amino acid. So citrulline plus aspartate amino acid using the synthase enzyme arginosuccinate and an energy source to make this arginosuccinate substrate. This arginosuccinate broken down arginosuccinase enzyme to give fumarate, which goes into TCA cycle as energy source, and it gives arginine. So arginosuccinate give fumarate, arginosuccinate give arginine, which is converted into urea and excreted from the body. So this is whole a brief elaboration for urea cycle, where we uses an amino acid and ammonia to get rid of that ammonia from the body by transferring it into ornithine, citrulline, and arginosuccinate. So if we have a deficiency or problem with ornithine transcarbamylase enzyme, so there is no transport of this ornithine carbamyl phosphate. So if there is no transport of this, there is a buildup of hyperammonemia. So it is a XLR condition, X-linked recessive disease. So no citrulline form, increase carbamyl phosphate, which leads to which gives a hyperammonemia state. So there is no citrulline, increase carbamyl phosphate, and it is causing the problem known as hyperammonemia. What will happen with this hyperammonemia? So alpha ketoglutarate add ammonia to make glutamate product. This glutamate, physiologically it is either converted into GABA using B6 or it is converted into glutamine. So whenever there is excess ammonia, all the glutamate is shifted towards the formation of glutamine, and there is less shifting of glutamate to GABA or B6. So this glutamine is causing osmotic shift in the brain tissue and causes cerebral edema. Also, if there is cerebral edema, it can causes pressure on the whole brain area and causing asterixis, slurring of speech, and also vision abnormality. So how to treat? We just need to focus on the conversion of this excess ammonia into another pathway. So we use lactulose, which acidify the gut and trap that ammonia. Also we use rifaximin and neomycin to kill ammonia producing bacteria, and also we restrict protein in the body. Neomycin and also restrict amino acid usage in the diet. So this is all about the urea cycle and essential amino acids and their derivatives.